You were told that the history of life on Earth is a story of evolution from simple to complex. From bacteria to fish, from fish to amphibians, from amphibians to reptiles, from reptiles to mammals, from mammals to us. Progress, direction, meaning. It is a beautiful story, and it covers approximately 12% of the actual time life has existed on Earth. The other 88%, the Precambrian, life was doing things that do not fit this framework at all. It was inventing chemistry that has no equivalent in the modern world. It twice froze the entire planet solid. It created organisms with symmetries found in no living creature today. And it did all of this without a single organism that had anything resembling a brain. 88% of the history of life is territory for which we barely have words. Today we explore it. Before we continue, please subscribe to this channel. Your support makes this work possible, and there is much more to explore. Thank you. Let us start with the numbers, because the numbers are necessary for understanding the scale of the problem. The Earth formed approximately 4.5 billion years ago. Life appeared, based on the earliest chemical evidence we can read from rocks, somewhere between 3.8 and 4.1 billion years ago. The Cambrian period, when animals with hard parts and complex body plans suddenly appear in the fossil record in dramatic abundance, began approximately 540 million years ago. The Precambrian, therefore, spans roughly 4 billion years. The Cambrian to the present spans 540 million years. The ratio is approximately 7:1. For every 1 year of the complex animal life we associate with evolutionary history, there were 7 years of Precambrian life doing something else entirely. This is not merely a quantitative difference, it is a qualitative one. The Precambrian was not a waiting period, a long geological hallway that life walked through on its way to the interesting rooms of the Cambrian and beyond. It was the main event. More fundamental biological innovations occurred during the Precambrian than in all subsequent time combined. More extreme planetary transformations occurred during the Precambrian than in all subsequent time combined. The Earth of the early Precambrian was not a younger version of the modern Earth. It was a different planet, running on different chemistry, inhabited by life that operated on principles we have only partially reconstructed, and that were not simply inferior versions of modern life's principles, but genuine alternatives, roads not taken that turned out to be viable for billions of years before being abandoned or overwhelmed. The very beginning, the Hadean, 4.5 to 4 billion years ago, almost certainly had life by its end, though the evidence is fragmentary and contested. The planet at this time was bombardment-scarred, volcanically violent, covered in an ocean that was hot and chemically reducing, under a sky with no oxygen and no ozone layer. The ultraviolet radiation reaching the surface from the young sun was intense enough to destroy the biological molecules of any modern organism within minutes of exposure. Whatever first lived in the Hadean Ocean had to contend with this environment, not as an exceptional challenge to be endured, but as the normal condition of existence. The oldest unambiguous evidence for life comes from the Archaean eon. the geological eon that spans approximately 4 to 2.5 billion years ago. In rocks from this period in Western Australia, South Africa, and the Arctic regions of Canada, geologists have found stromatolites. These are layered structures, typically domed or columnar, built by communities of microorganisms, primarily photosynthetic bacteria, though the Archaean versions were more diverse in their metabolic strategies than any modern stromatolites. The individual organisms that built them are invisible to the naked eye. The structures they built are not. A stromatolite community can extend for miles, forming what amounts to a microbial reef. A reef not of corals and sponges as in modern oceans, but of bacteria building layered mineral structures that can be feet or meters in height. Looking at a field of Archaean stromatolites, which you can do in a few places on Earth where these ancient rocks are exposed, most notably at Shark Bay in Western Australia, where modern stromatolites still grow in hypersaline conditions very similar to their ancient predecessors, is perhaps the closest you can come to experiencing the Precambrian directly. The structures are clearly biological. They have the characteristic domed shape, the layered internal structure, the relationship to the ancient seafloor. But the biology that built them has almost nothing in common with the biology of visible organisms. No cells with nuclei, no multicellular organization, no tissues, no organs, no differentiation between different types of cells. Just communities of single-celled bacteria, each organism doing everything for itself, cooperating only in the most minimal chemical sense with its neighbors. And the ocean those stromatolites grew in was genuinely alien by modern standards. The Archaean Ocean was iron-rich. Dissolved ferrous iron was present in concentrations far exceeding anything in modern seawater, giving the ocean a greenish or yellowish tint in some areas. The water was warm, surface temperatures significantly higher than today, maintained by a thick greenhouse atmosphere of carbon dioxide and methane, even though the sun was dimmer than its current output. There was essentially no free oxygen in the atmosphere or dissolved in the surface ocean. The dominant gas was nitrogen, with significant carbon dioxide and methane and various sulfur compounds. The sky was not blue. It was probably some shade of orange or pale amber from the atmospheric chemistry, with the methane haze that microbial methanogens were continuously producing, creating a smog layer in the upper atmosphere, similar in some ways to what we observe on Saturn's moon Titan today. In this world, this orange-skied, iron-green ocean, oxygen-free, hot world, life was extraordinarily diverse at the metabolic level, even while it was completely uniform at the morphological level. Every organism was a single cell, but those single cells were running metabolisms that are mostly absent from the modern world. Metabolisms based on sulfur, organisms that reduced sulfate to hydrogen sulfide for energy, or oxidized hydrogen sulfide for energy. Metabolisms based on iron, organisms that reduced ferric iron to ferrous iron, or the reverse. Metabolisms based on various nitrogen compounds. Metabolisms based on hydrogen. The Archaean Ocean was not a simple place with simple chemistry. It was an extraordinarily complex chemical soup in which dozens of different microbial metabolic strategies were operating simultaneously, each occupying a slightly different chemical niche, each producing waste products that became the food source for another metabolic strategy. This is the architecture of the invisible world. Not one type of life, but a dense web of interacting microbial metabolisms, cycling elements through the ocean and atmosphere with a throughput and sophistication that we are only beginning to appreciate. The carbon cycle, the sulfur cycle, the nitrogen cycle, the iron cycle, all of these fundamental biogeochemical cycles that keep the modern Earth habitable were being operated by this invisible microbial world 3.5 billion years ago, with a precision and a stability that is genuinely remarkable given the apparent simplicity of the individual organisms involved. No brain coordinated this. No organism planned it. It was the emergent property of billions of trillions of single-celled organisms, each doing what their chemistry compelled them to do, and the combined effect being a regulated, self-sustaining chemical system that kept the planet in a habitable state for billions of years. Then, at some point between 2.7 and 2.4 billion years ago, something appeared that would eventually destroy this entire system: Cyanobacteria. The photosynthetic revolution that these organisms brought was not an improvement on what existed. From the perspective of the existing Archaean biosphere, it was a catastrophe. Cyanobacteria performed oxygenic photosynthesis. They split water molecules using sunlight, extracting hydrogen for carbon fixation and releasing molecular oxygen as a waste product. Oxygen, to the anaerobic organisms that had dominated the Archaean biosphere for over 1 billion years, was not a resource. It was a poison, highly reactive, chemically aggressive, destructive of the organic molecules and metabolic enzymes that anaerobic life had spent billions of years perfecting. The oxygen that cyanobacteria began releasing into the Archaean Ocean was, from the perspective of most of the life then existing, an environmental catastrophe of the first order. The Great Oxidation event, the dramatic accumulation of free oxygen in Earth's atmosphere beginning approximately 2.4 billion years ago, is sometimes described in evolutionary accounts as an opportunity. Free oxygen enabled aerobic respiration, which extracts far more energy per unit of food than anaerobic processes, enabling the evolution of more complex, more energetic, more active organisms. All of this is true. But the immediate effect was mass extinction on a scale that dwarfs anything in the subsequent fossil record. The anaerobic world that had existed for 2 billion years was, in a geological sense, rapidly dismantled. Organisms that had no defenses against oxidation found themselves in a world that was becoming lethally oxidizing. The metabolic strategies that had sustained life for billions of years became untenable in an environment where oxygen was present. The majority of the biomass and biodiversity of the Archaean world was eliminated. The geological evidence for this catastrophe is written in rock in a form that is both dramatic and permanent. The banded iron formations, those distinctive alternating layers of iron-rich and iron-poor sediment, that today are mined as iron ore across multiple continents, record the progressive oxidation of the Archaean Ocean's dissolved iron. For hundreds of millions of years, as cyanobacteria produced oxygen, That oxygen was immediately absorbed by the dissolved ferrous iron in the ocean, oxidizing it to ferric iron, which is insoluble and precipitated to the ocean floor. The banded structure reflects seasonal or cyclic variation in this oxidation process. Dark iron-rich layers, lighter silica-rich layers, repeating for hundreds of millions of years. When the banded iron formations finally stop, when this distinctive rock type disappears from the geological record approximately 1.8 billion years ago, it signals that the ocean's iron buffer has been exhausted. There is no more dissolved iron to absorb the oxygen. From this point, oxygen begins to accumulate in the atmosphere in earnest. The atmosphere that emerged from the Great Oxidation Event was chemically transformed in ways that affected the entire planet. The methane that had been a major component of the Archaean atmosphere was destroyed by reaction with oxygen. The loss of methane, a potent greenhouse gas, dramatically reduced the greenhouse warming that had been keeping the planet warm despite the dimmer early sun. The result was global cooling of a severity that is difficult to overstate. The Huronian glaciation, approximately 2.4 to 2.1 billion years ago, was the first of the Precambrian Snowball Earth episodes, periods in which ice sheets extended from the poles all the way to the equator, covering the entire planet's surface in ice. The evidence for Huronian glaciation is found on every continent in the form of ancient glacial deposits. Distinctive rock types like dropstones, which are boulders dropped by melting icebergs into fine-grained sediments, and striated pavements, rock surfaces scratched by glacial ice movement, at geological locations that were unambiguously in the tropics at the time of their formation. Ice at the equator, ice covering the entire ocean surface, ice everywhere. How thick was this ice? Estimates vary, but in the full snowball scenario, the ocean ice may have been a kilometer or more thick across most of the planet's surface. The albedo, the reflectivity of ice, is dramatically higher than that of open ocean or bare rock. An ice-covered planet reflects most incoming sunlight back into space rather than absorbing it as heat. The positive feedback loop is terrifying in its logic. Cooling begins. Ice forms. Higher albedo reflects more heat. More cooling occurs. More ice forms. Once started, this feedback loop can drive the planet to complete glaciation remarkably quickly in geological terms, perhaps within tens of thousands of years. Getting out of it is a different problem entirely. Life survived the Huronian glaciation. We know this because life exists today. But how it survived in a planet that was, at least at the surface, completely frozen is a genuinely interesting question. The most likely answer involves two types of refugia: hydrothermal systems on the seafloor, where the heat from the Earth's interior maintained liquid water and chemical energy regardless of surface conditions, and small open water areas near volcanic islands, or deep ocean vents where the ice was thin or absent. Life did not thrive during the Huronian snowball. It retreated to the margins of the possible and waited. The exit from the snowball state came through the same volcanism that had been operating throughout the glaciation. Volcanoes do not stop when the surface freezes. They continued to emit carbon dioxide throughout the glaciation. But with the ocean frozen and the silicate weathering that normally removes carbon dioxide from the atmosphere dramatically reduced by the absence of liquid water on the land surface, the volcanic carbon dioxide accumulated in the atmosphere without being neutralized. Over millions of years, perhaps tens of millions of years, the greenhouse effect of this accumulated carbon dioxide was sufficient to overwhelm the albedo effect of the ice, triggering rapid deglaciation. The melting, when it finally began, may have been extremely rapid by geological standards, from fully glaciated to deglaciated in perhaps a few thousand years. The immediate aftermath of snowball deglaciation was a world of extreme chemical disequilibrium. Hot, chemically aggressive, with the ocean receiving enormous inputs of dissolved minerals from the suddenly thawed continental surfaces. This is important context for what happened next, or rather for what happened approximately 600 million years later, when another, even more extreme Snowball Earth episode fundamentally transformed life on this planet. Because the Precambrian had not one snowball event, but at least two, possibly more, and the later ones, the Sturtian and Marinoan glaciations of approximately 717 to 635 million years ago, appear to have been even more complete than the Huronian. The evidence for these later snowball episodes is more precisely preserved and more extensively studied than the Huronian evidence. And it points to ice extending to within a few degrees of the equator, perhaps all the way to the equator, for millions of years. What happened in the aftermath of the Marinoan glaciation, the most recent and most severe of the confirmed snowball episodes, is where the Precambrian story becomes most directly connected to everything that followed. The rapid deglaciation produced a world of extraordinary chemical richness. Minerals that had been locked in permafrost and glacial ice for millions of years were suddenly dissolved and entering the ocean in enormous quantities. Nutrients, phosphorus especially, a key limiting nutrient for biological productivity, were available in concentrations far higher than anything the pre-glacial ocean had seen. The post-snowball ocean was, in chemical terms, supercharged. Into this chemically supercharged post-snowball world, approximately 635 million years ago, life entered the Ediacaran period. And the Ediacaran is where the Precambrian stops being merely ancient and strange and becomes genuinely unsettling in its implications. The Ediacaran organisms, first discovered by a young Australian geologist named Reginald Sprigg in 1946, sitting on a rock in the Ediacara Hills of South Australia eating his lunch, were not what anyone expected Precambrian life to look like. The rocks they came from were Precambrian rocks, which at the time were supposed to contain nothing visible. Sprigg found impressions in the rock the size of dinner plates, circular, oval, some with internal complexity, ribbing, symmetry, structure. They were clearly biological, they were clearly large, and they were clearly unlike anything alive today or previously known from the fossil record. What were these things? After 70 years of intensive study, the honest answer is, we are not entirely sure. We know they were multicellular. Their size and the structural complexity of their impressions makes a single-celled interpretation untenable. We know they were soft-bodied. They left no mineralized remains, only impressions in fine-grained sediment that preserves their external form, but tells us frustratingly little about their internal organization. And we know that several of the major Ediacaran taxa, the groups of organisms with distinctly recognizable body forms, do not clearly fit into any of the major animal phyla that appear in the Cambrian and persist to the present. Dickinsonia is the most famous example. This organism, which ranges in size from a few millimeters to over a yard in length, has a distinctly oval form with a series of parallel segments running along its length, separated by a midline. It looks, at first glance, like a very flat, very large organism in the worm family. But the segments are not paired across the midline as they are in segmented worms and other bilaterally symmetric animals. They are offset, with each segment on one side positioned between segments on the other side. The internal organization implied by this arrangement does not match any modern animal. And when researchers analyzed the lipid chemistry preserved in Dickinsonia fossils, specific fat molecules that can survive in rock for hundreds of millions of years, they found cholesterol-related compounds, which firmly places Dickinsonia in the animal kingdom. Dickinsonia was an animal, but it was an animal with a body plan found in no living or previously known fossil group. It was not a worm. It was not a flatworm. It was not an echinoderm or a mollusk or any of the other major animal groups. It was something that evolution tried, made successful for tens of millions of years, and then completely abandoned when the Cambrian began. Tribrachidium is even stranger. This is a disc-shaped organism with threefold symmetry, three identical units arranged around a central axis, each separated from the others by 120 degrees. Look at any animal alive today. Find me threefold symmetry. You cannot. It essentially does not exist in the animal kingdom. Bilateral symmetry, the left-right mirror image that characterizes most animals, including humans, is everywhere. Five-fold symmetry exists in echinoderms. Various forms of radial symmetry exist in jellyfish and corals. But threefold? Tribrachidium had it. A stable, consistent, repeated body plan based on an organizational principle that the animal kingdom has not used since. This is not a transitional form to something with two-fold or four-fold symmetry. It is a stable expression of a symmetry rule that exists nowhere else in the entire subsequent history of multicellular life. Charnia was found in England in 1957 by Roger Mason, who was 15 years old at the time and rock climbing in Charnwood Forest when he spotted it in rocks that geologists had confidently declared too old to contain complex fossils. It looks like a frond, a branching, leaf-like structure attached to the substrate by a disc-shaped holdfast, with branches arranged in a herringbone pattern on either side of a central stem. It looks unmistakably like a plant. But Charnia fossils have been found in rocks representing water depths where no photosynthesis was possible. It was not a plant. It was a sessile, permanently attached organism that lived on the ocean floor in depths where no sunlight reached, obtaining nutrition by some method that did not involve photosynthesis. The current leading hypothesis is osmotrophy, absorbing dissolved organic molecules directly from the surrounding water through the body surface. A strategy that works only if the surrounding water contains significantly more dissolved organic material than modern seawater does. The Ediacaran Ocean was apparently rich enough in dissolved organics to support large, complex organisms feeding this way. No modern ocean is. Pteridinium is perhaps the most alien of all. This organism, which could reach nearly a foot in length, has a frond-like form but with a distinctive three-veined structure. 3 planar wings arranged around a central axis, like a propeller with 3 blades. Each wing shows a repetitive internal structure of smaller units arranged in rows. The entire organism, in its 3-dimensional reconstruction, looks like nothing that has ever lived before or since. Not a plant, not an animal, not a fungus. Something that built a large, complex, organized body using a structural principle that has no analog in the modern world. Whatever pteridinium was, it was clearly successful. It is found in multiple geological localities across several continents, suggesting it was widespread and abundant. And then it disappears entirely from the fossil record at the Ediacaran-Cambrian boundary, leaving no descendants anyone has been able to identify. What were the Ediacaran organisms eating? This question is more profound than it might initially appear. Modern complex animals, all of them without exception, obtain energy either through photosynthesis or by consuming other organisms, or by decomposing dead organic material. These are the 3 basic nutritional strategies available to complex life. The Ediacaran organisms seem to have been using none of these in quite the way modern organisms use them. The osmotrophic hypothesis, direct absorption from surrounding water, requires an ocean chemistry that no longer exists. Some researchers have proposed that the flat, disc-like body forms of organisms like Dickinsonia maximized the surface area available for this kind of passive absorption. Others have proposed that at least some Ediacaran organisms harbored symbiotic photosynthetic or chemosynthetic microbes in their tissues, the way modern corals harbor symbiotic algae, getting their nutrition through internal biological chemistry rather than active feeding. The absence of mouths is striking. Among the soft-bodied impressions preserved from Ediacaran fauna, clear evidence for a mouth, an opening through which food was actively ingested, is rare and disputed. Most Ediacaran organisms appear to have had no mouth in the modern sense. They had no gut, no tube through which ingested food could pass and be digested. No circulatory system to distribute nutrients from a gut to the rest of the body. They were, in the fundamental sense that modern animal anatomy assumes, not animals at all in the way we normally think about animals. They were large, complex, multicellular, with internal organization and bilateral or other symmetry. But they operated without the basic digestive machinery that we associate with animal life. Here is the thing that puts all of this in perspective. The Ediacaran period lasted from approximately 635 to 540 million years ago, roughly 95 million years. The time between the Ediacaran's end and the present is approximately 540 million years. The organisms that dominated the Ediacaran, Dickinsonia, Charnia, Pteridinium, and dozens of other genera, persisted and diversified for longer than the entire span of time from the Cambrian explosion to today. Whatever they were doing it successfully for an enormous stretch of time. They were not proto-animals struggling towards some more complex future. They were the complex life of their time, perfectly adapted to the Ediacaran world, ecologically dominant, widespread, and diverse. And then they disappeared. In the geological record, the transition from the Ediacaran fauna to the Cambrian fauna is abrupt. Abrupt in geological terms, meaning it happened within a few million years, rather than over tens of millions. The Ediacaran organisms largely vanish from the fossil record as the Cambrian begins. The Cambrian organisms, with their shells, their eyes, their complex appendages, their clearly recognizable relationship to modern animal phyla, appear seemingly from nowhere in abundance. What killed the Ediacaran world? Several hypotheses exist. The leading one involves the animals themselves, specifically the appearance of animals with mouths and guts that could actively eat other organisms. The first predators. In the Ediacaran world, there appear to have been no predators in the modern sense, no organism that actively captured and ate other organisms. The entire ecosystem was, in a sense, peaceful. Organisms absorbed nutrients from their environment passively, competed for light or dissolved organics or substrate space, but did not eat each other. The appearance of the first true predators would have been catastrophic for organisms that had evolved for 95 million years in a predator-free world. A Dickinsonia that spent its entire evolutionary history in an environment with no active predation would have no defenses against an organism that could simply bite it or engulf it. The appearance of even primitive predatory behavior would have given the new organisms an enormous advantage and would have driven rapid selection pressure for shells, armor, speed, all the features we see appearing in the Cambrian. There is another factor: bioturbation. The burrowing of animals through sediment, the mixing and churning of the seafloor by organisms digging for food or shelter, was absent or minimal in the Ediacaran. The microbial mats that covered the Ediacaran seafloor, stabilizing the sediment and providing a substrate for the attached sessile organisms, would have been destroyed by burrowing. When burrowing organisms appeared at the Ediacaran-Cambrian transition, they literally tore apart the ecological foundation that the Ediacaran community was built on. The stable, mat-covered seafloor that had persisted for hundreds of millions of years was churned into loose sediment. Organisms that depended on mat attachment could not persist. But here is the deepest puzzle that the Precambrian poses for our understanding of life. The Cambrian explosion, the rapid appearance of all the major animal body plans that persist to the present, did not come from nowhere. The genetic machinery required to build complex animal bodies does not arise spontaneously. The HOX genes, the toolkit genes that control body plan development in all modern animals, must have been present in some ancestral form before the Cambrian. The complex cell signaling pathways that coordinate multicellular development were not invented in the Cambrian. They must have been assembled during the Precambrian in organisms that left little obvious trace in the fossil record. This means there is a hidden chapter, a long period during the late Precambrian when genetic complexity was accumulating in organisms that were still morphologically simple. Animals that were genetically sophisticated but physically unremarkable. Organisms that had the potential for the Cambrian explosion encoded in their DNA, but had not yet expressed it in their body plans. The Cambrian explosion, in this view, was not the invention of new biology. It was the expression of biology that had been building for hundreds of millions of years in organisms we cannot see in the fossil record. The molecular clock approach, using the rate of accumulation of genetic mutations to estimate when lineages diverged, suggests that many of the major animal lineages diverged from each other long before the Cambrian. Some estimates place the divergence of the major animal phyla as early as 800 to 1,000 million years ago, well into the Precambrian, long before any of these lineages left clear body fossils. If this is correct, then the animals of the Cambrian were not new creatures, but ancient lineages that had been present in small, soft-bodied, cryptic forms for hundreds of millions of years, before suddenly adopting the large, hard-bodied, ecologically dominant forms that fill the Cambrian fossil record. The trigger for this transition, for the shift from small and cryptic to large and prominent, is itself debated. Rising oxygen levels in the aftermath of the Merino and Snowball glaciation is the leading candidate. Complex animals require more oxygen than simple ones because they have more tissue to oxygenate at greater distances from any body surface. The progressive oxygenation of the deep ocean during the Ediacaran period, which is well documented in the geochemical record, may have crossed a threshold that allowed larger, more active, more metabolically demanding body plans to become viable. The Snowball Earth events, which disrupted existing ecosystems and delivered enormous pulses of nutrients to the post-glacial ocean, may have provided the ecological opportunity for the new body plans to be expressed. The appearance of predation created the selection pressure for the defensive structures, shells, armor, spines, that make complex animals visible in the fossil record. All of these factors, oxygen, nutrients, predation, ecological opportunity, may have converged in the Ediacaran-Cambrian transition to produce the most dramatic biological radiation in the history of animal life. The Cambrian explosion was not a mystery in the sense of having no explanation. It had too many explanations, each partially valid, each contributing to a process that was the culmination of 4 billion years of Precambrian biological innovation. The oldest bacteria are with us still. They have not been replaced by something fundamentally better. They have been joined by more complex life while continuing to do what they have always done. The microbial world of the Precambrian did not end with the Cambrian explosion. It continues today. The soil beneath your feet contains bacteria performing metabolisms that differ little from the metabolisms of Archaean organisms 3.5 billion years ago. The hydrothermal vents on the ocean floor host microbial communities that are the closest living analogues to the communities of the early ocean. The anaerobic bacteria in the digestive systems of every complex animal, including every human being, are doing anaerobic chemistry that dates to before the Great Oxidation event. The Precambrian is not past, it is present. The invisible world that predates all visible life is still the foundation on which everything visible rests. Consider what the Precambrian did for us, specifically. Without the Great Oxidation event, there would be no free oxygen in the atmosphere. Without free oxygen, no ozone layer. Without an ozone layer, intense ultraviolet radiation reaching the land surface makes complex land life essentially impossible. The terrestrial biosphere, every plant, every land animal, everything that lives on the continents, owes its existence to the cyanobacteria of the Archaean and Proterozoic oceans, which spent billions of years producing the oxygen that made the ozone layer possible. Without the Snowball Earth events, the chemical supercharging of the post-glacial ocean may never have occurred. Without that supercharging, the nutrients required to fuel the Ediacaran biological expansion may not have been available. Without the Ediacaran expansion, the genetic and ecological groundwork for the Cambrian explosion may not have been laid in the way it was. The cascade of dependencies runs backward through time, each event in the Precambrian necessary for the next, the whole sequence necessary for what we recognize as complex life to appear at all. And there is one more thing the Precambrian tells us that is worth sitting with. Life is extraordinarily persistent. The Huronian snowball froze the planet. Life survived. The Great Oxidation event poisoned the atmosphere for the majority of life then existing. Life adapted and continued. The Marinoan snowball froze the planet again, more completely, for longer. Life survived again. Whatever the Precambrian threw at life, and it threw extraordinary things, life did not stop. It retreated, contracted, found refugia, waited, and then expanded again when conditions improved. The resilience is not inspirational in the motivational sense. It is just chemistry, just the property of self-replicating systems, that the ones capable of surviving pass their chemistry forward, while the ones that cannot do not. But the cumulative result is 4 billion years of biological continuity in the face of conditions that would strike any modern observer as incompatible with life. We are the product of that continuity. Every cell in your body contains molecular machinery, the ribosome, the genetic code, the ATP synthase, that was assembled in the Precambrian and has been passed forward, modified but never replaced, through every organism in the unbroken chain of biological descent, from the first life to you. The Precambrian is not prehistory in the sense of being irrelevant to the present. It is the foundation on which everything present stands. The 88% that the textbooks mostly skip is the part that built the world you inhabit. The story of the Precambrian is not the story of life preparing to become interesting. It is the story of life being interesting in ways we are still learning to recognize. Threefold symmetry. organisms without mouths feeding on dissolved ocean chemistry, a planet freezing solid and thawing catastrophically, bacteria poisoning the atmosphere with oxygen and accidentally enabling the evolution of everything complex that followed. A world that looked, from orbit, like nothing currently in the solar system. Orange sky, green, iron-rich ocean, no land life, no animal sounds, no complexity visible from any distance greater than the microscopic scale. That world was real. It lasted 4 billion years. It ended not with a bang, but with a slow transformation whose final act, the Cambrian explosion, is dramatic enough that we often mistake it for a beginning. It was not a beginning. It was the conclusion of the longest, strangest, most consequential story in the history of life on Earth. And we are its last chapter, still being written, still carrying in our cells the molecular heritage of that invisible world that built everything we are. The next time you look at a clear night sky and see the stars, consider that the oxygen you are breathing to see them was produced by organisms that no human has ever seen, living in an ocean that no longer exists, on a planet that looked nothing like the one you are standing on. The Precambrian gave you that breath. It gave you everything. Let us go deeper into the chemistry of the Archaean Ocean, because it is one of the most alien environments in the history of this planet, and understanding it changes how we think about what life can be. The Archaean Ocean was not merely different from modern seawater in composition. It was operating under fundamentally different chemical rules, supporting fundamentally different biological strategies, cycling elements in ways that have no precise modern analog. Take the sulfur cycle. In the modern ocean, sulfur exists primarily as sulfate, the oxidized form, because the oxygen-rich modern ocean has oxidized most of the reduced sulfur compounds that enter it from volcanoes and other sources. In the Archaean Ocean, with essentially no free oxygen, sulfur was present in reduced forms: hydrogen sulfide, elemental sulfur, various sulfide minerals. This made sulfur a usable energy source in a way it is not in the modern ocean. Sulfur-reducing bacteria could gain energy by using sulfate as an electron acceptor, reducing it to hydrogen sulfide. Sulfur-oxidizing bacteria could gain energy by doing the reverse, using the chemical potential between the reduced and oxidized sulfur compounds. This sulfur-based energy economy, running in the absence of oxygen, was one of the major metabolic engines of the Archaean Ocean. The iron cycle was similarly transformed. Modern seawater contains almost no dissolved iron. It is present in trace amounts, and its scarcity is actually a major limiting factor for biological productivity in much of the modern ocean. In the Archaean Ocean, dissolved ferrous iron was abundant, contributing to the greenish or brownish appearance of the water in some areas. Iron-reducing bacteria could use ferric iron compounds as electron acceptors, gaining energy while producing ferrous iron. Iron-oxidizing bacteria could do the reverse. The iron cycle, like the sulfur cycle, was a living metabolic engine sustained by the chemical gradient between the oxidized and reduced forms of the element. What is extraordinary about these Archaean metabolisms is not merely their antiquity, but their effectiveness. The organisms running these sulfur and iron cycles were not inefficient. struggling ancestors of more sophisticated modern life. They were optimized by billions of years of selection to extract the maximum available energy from their chemical environments. Some of them ran these metabolisms at efficiencies close to the thermodynamic maximum, the theoretical limit imposed by the laws of physics. In this narrow sense, they were not more primitive than modern organisms. They were as refined and efficient as any life has ever been, at the specific task of converting available chemical energy into biological work. The methanogens deserve special attention. Methane-producing archaea, the methanogens, were among the most ecologically important organisms of the Archaean world. They produced methane as a metabolic byproduct, and this methane accumulated in the atmosphere to a degree that had profound effects on the planetary climate. The faint young sun problem, the paradox that the early sun was significantly dimmer than today, and yet the early Earth was not frozen, is most plausibly resolved by the presence of methane in the Archaean atmosphere as a potent greenhouse gas. The methanogens, by producing methane, were not merely running their own metabolisms. They were regulating the temperature of the planet, keeping it warm enough for liquid water and for the rest of the biosphere to function. The most important climate regulators of the first 2 billion years of life's history were invisible microbes producing the simplest of small molecules. When Cyanobacteria began producing oxygen, one of the first atmospheric consequences was the destruction of this methane greenhouse. Oxygen reacts with methane. The reaction is not instantaneous at atmospheric temperatures, but over geological timescales. Rising oxygen levels progressively depleted the methane that methanogens were producing. This is what triggered the Huronian glaciation. The methanogens, by their metabolism, had been maintaining a warm planet for billions of years. When their methane was chemically destroyed by the new oxygen, the warming effect diminished, and the planet froze. The largest organisms to have ever caused a global ice age were microscopic. Now let us go to the eukaryotes, the cells with nuclei that eventually gave rise to all complex life, including ourselves, because the story of their origin in the Precambrian is one of the most remarkable in biology. All organisms more complex than bacteria are eukaryotes. Every cell in your body is a eukaryote. Every plant cell, every fungal cell, every protist cell. The defining feature is the nucleus, a membrane-bounded compartment within the cell that contains the genetic material, separating it from the rest of the cell's chemistry. Eukaryotes also have mitochondria, organelles that perform aerobic respiration, extracting energy from food using oxygen. And plant and algal cells have chloroplasts, organelles that perform photosynthesis. Mitochondria and chloroplasts are, as has been established beyond reasonable scientific doubt, descended from bacteria that were engulfed by ancient eukaryotic cells and retained as endosymbionts rather than being digested. The evidence is comprehensive. Both mitochondria and chloroplasts have their own DNA, distinct from the nuclear genome, and this DNA resembles bacterial DNA rather than eukaryotic nuclear DNA. Both divide by binary fission, like bacteria, rather than by the mitotic cell division used by eukaryotic cells. Both have a double membrane structure consistent with an origin as an engulfed bacterium. The protein synthesis machinery of both organelles is bacterial in character rather than eukaryotic. The mitochondrial ancestor was almost certainly a member of the alpha Proteobacteria, a group of bacteria that includes the modern Rickettsia, the cause of typhus, and Brucella, the cause of brucellosis. The chloroplast ancestor was a cyanobacterium, specifically a member of the group responsible for the Great Oxidation event. This means that every cell in your body contains the descendants of a bacterium that was engulfed by an ancestral cell somewhere between 1.5 and 2 billion years ago and never digested. The organism that would become your mitochondria was once a free-living bacterium in the Proterozoic ocean. It has been inside cells, doing the same basic chemistry of aerobic respiration ever since. The timing of eukaryote evolution in the Precambrian is critical to understanding the Ediacaran-Cambrian transitions. The earliest unambiguous eukaryotic fossils appear in rocks approximately 1.8 billion years old. Single-celled organisms with nuclei, clearly distinct from bacteria, preserved in fine-grained Proterozoic sediments. These early eukaryotes were, for most of the next billion years, single-celled. They diversified into many lineages. The ancestors of modern algae, fungi, and animals all diverged from a common eukaryotic ancestor during this period. But for hundreds of millions of years after the acquisition of mitochondria, eukaryotes remained microscopic single cells. The nuclear genome was growing, the metabolic capabilities were diversifying, the genetic toolkit for multicellular development was presumably being assembled, but none of this complexity was expressed in visible body plans. The gap between the first eukaryotes at 1.8 billion years and the first unambiguous multicellular animals at approximately 600 million years is over 1 billion years. This interval, sometimes called the boring billion by geologists, a term that is simultaneously accurate and profoundly misleading, appears to have been a period of relative biological stasis at the organismal level, but extraordinary biological innovation at the molecular and cellular level. The genome was evolving. Cell signaling pathways were being elaborated. The regulatory networks that control gene expression in development, the networks that allow a single fertilized egg to produce, through carefully regulated cell division and differentiation, an organism with hundreds of different cell types and complex three-dimensional organization, were being built gene by gene, interaction by interaction, during this apparently quiet billion years. What broke the stasis? Multiple factors converged at the end of the Proterozoic. The Sturtian and Marinoan snowball glaciations disrupted existing ecosystems and created post-glacial conditions that strongly favored new metabolic and ecological strategies. Atmospheric oxygen continued to rise, eventually reaching levels sufficient to support larger, more metabolically active organisms. And possibly, this is a compelling hypothesis, a threshold was crossed in the molecular toolkit for development. The point at which sufficient genetic regulatory machinery had accumulated that multicellular development of genuine complexity became possible for the first time. The transition from single-celled to multicellular life appears to have happened independently multiple times in different lineages. Animals evolved multicellularity once from a single common ancestor. Plants evolved multicellularity independently from different single-celled ancestors. Fungi evolved multicellularity independently. Various algal lineages evolved it multiple more times, and numerous attempts, the colonial green algae, the cellular slime molds, various other organisms that form multicellular bodies but never evolved the complexity of true multicellularity, have been happening throughout eukaryotic evolutionary history. Multicellularity, far from being a rare accident, appears to be a solution that evolution finds repeatedly whenever the genetic prerequisites are in place and the ecological opportunity exists. The Ediacaran period represents the ecological expression of this genetic potential, built up through hundreds of millions of years of Precambrian molecular evolution. The large, complex, soft-bodied organisms of the Ediacaran were not simple experiments on the road to something better. They were the first full flowering of multicellular animal life, using the genetic toolkit that had been assembled during the boring billion to build large, organized, ecologically diverse bodies. That many of these body plans did not persist into the Cambrian does not make them failures. They were successful for 95 million years in the conditions of the Ediacaran Ocean. The conditions changed, and the body plans that had been tuned to those conditions could not adapt rapidly enough. The fossil record of the Precambrian has a fundamental preservation problem that must be understood to appreciate how limited our knowledge actually is. Fossilization requires that an organism or its hard parts be buried rapidly in sediment and then preserved through the subsequent geological processing of that sediment, a process that typically takes hundreds of millions of years and involves significant transformation of the original material. Soft-bodied organisms, organisms without shells, bones, or other mineralized hard parts, are fossilized only under exceptional circumstances, when burial is rapid enough and sediment chemistry favorable enough to preserve impressions or organic material before decomposition destroys it. The Ediacaran organisms were soft-bodied. Their preservation in the rock record is exceptional by the standards of fossilization. The conditions in the Ediacaran Ocean, including the extensive microbial mats that covered the seafloor and retarded the decomposition of organisms buried in them, were unusually favorable for soft body preservation. Even so, what we have preserved is only a fraction of what existed. Body plans that did not happen to be preserved in the specific sedimentary environments that favor Ediacaran-style preservation are simply absent from the record. The Ediacaran fauna we know, Dickinsonia, Charnia, Pteridinium, and the roughly 50 to 60 genera that have been described, is almost certainly a highly incomplete sample of the actual Ediacaran community. For the 3 billion years of the Archaean and early Proterozoic, the preservation problem is even more severe. The primary evidence for Archaean life is not body fossils. There are no complex body fossils from this period, but chemical and isotopic signatures preserved in ancient rocks. carbon isotope ratios that indicate biological carbon fixation, sulfur isotope signatures that record microbial sulfur cycling, lipid biomarkers, specific organic molecules produced by particular types of organisms that can survive in rock for billions of years, morphological microfossils, microscopic structures that appear to be cell walls or other biological structures, but are notoriously difficult to distinguish from inorganic mineral formations of similar shape and size. The study of Archaean microbial life is, in a real sense, forensic science applied to the most ancient available evidence. Every conclusion is hedged with uncertainty. The oldest claimed body microfossils, structures from rocks in the apex chert of Western Australia dating to 3.5 billion years, described in 1993 as fossilized cyanobacteria-like filaments, have been contested by researchers who argue that the structures are of inorganic origin, produced by hydrothermal mineral precipitation rather than by biological activity. The debate is ongoing and has not been definitively resolved. What is not contested is the isotopic evidence for biological carbon fixation in rocks of approximately this age. The carbon isotope ratios preserved in Archaean organic carbon clearly indicate biological chemistry rather than inorganic processes. Life was there. Its physical form is uncertain. This uncertainty is not a failure of science. It is the honest acknowledgement of what 4 billion years of geological processing does to evidence. The oldest rocks on Earth, the Acasta gneiss of Canada, at 4.02 billion years old, are metamorphic rocks, meaning they have been subjected to heat and pressure sufficient to destroy most of the chemical signatures that would identify biological activity. The Jack Hills zircons of Australia, individual mineral crystals as old as 4.4 billion years, the oldest material on Earth, preserve geochemical information but no biological signatures. We are reading the most ancient chapters of life's history from vanishingly fragmentary evidence, and the conclusions we can draw are correspondingly limited. What we can say with reasonable confidence is this: life on Earth has been continuous for at least 3.5 billion years, possibly longer. For the first 3 billion of those years, life was exclusively or almost exclusively microbial. During that microbial era, life transformed the planet's atmosphere, regulated its climate, cycled its essential nutrients, and assembled the genetic complexity that would eventually produce visible organisms. The eukaryotic cell, the cell type from which all visible complex life is built, appeared approximately 1.8 billion years ago through the endosymbiotic incorporation of bacterial cells. Multicellular complex organisms appeared approximately 600 million years ago, following the extreme environmental disruption of the Marinoan snowball glaciation. And the Ediacaran fauna, the first large, diverse, ecologically complex animal community, populated the ocean for 95 million years before the Cambrian explosion reorganized terrestrial and marine ecosystems into the configuration from which the modern world is descended. Each of these events was a genuine biological revolution. None of them was inevitable. Each required a specific set of prior conditions, molecular, ecological, environmental, that themselves required the full sequence of prior events extending back to the origin of life itself. The Cambrian explosion was not a lucky accident that happened to produce complex animals on this particular planet. It was the outcome of 4 billion years of preparation, of chemistry being assembled molecule by molecule, metabolism by metabolism, cell by cell, organism by organism, into a system that was finally capable of the rapid diversification we see in the Cambrian fossil record. We are the result of this preparation, not the intended result. Evolution has no intentions, but the current result, the present state of a process that began before the oldest rocks on Earth were formed and has been running continuously ever since. The Precambrian is not our origin story in the simple sense of being interesting history. It is our foundation in the literal sense that the molecular machinery of our cells, the oxygen we breathe, the nitrogen in our proteins, the phosphorus in our DNA, all of these were processed and made biologically available by Precambrian organisms in Precambrian environments. The next time you see a clear night sky, the fact that you can see it at all is a Precambrian gift. The oxygen in the air, the ozone in the upper atmosphere, blocking the ultraviolet that would otherwise make the surface lethal to complex life. All of it was produced by Cyanobacteria that lived and died billions of years before your most ancient mammalian ancestor was born. The Precambrian is not behind us. It is underneath us in every sense. There is a specific place on Earth where the Precambrian is not merely a subject of scientific study, but an immediate physical presence, where you can walk across it, sit on it, run your hand over the surface of a world that is 3.5 billion years old. The Pilbara region of Western Australia contains some of the oldest exposed rocks on the planet's surface. The landscape is austere, red-brown rock, minimal vegetation, an ancient craton that has been stable and exposed for an extraordinary span of geological time. When you look at the surface of the Pilbara's ancient cherts and basalts, you are looking at material that was here before the first eukaryotic cell existed, before the first Snowball Earth event, before the Great Oxidation event. The ground itself predates the oxygen you are breathing. In certain formations within the Pilbara, the Dresser Formation at Pilbara's northern extent, approximately 3.5 billion years old. Geologists have found the oldest well-preserved stromatolites in the world. The structures are preserved in chert, a fine-grained silica rock that forms when silica-rich waters mineralize organic material rapidly, creating a permanent record in stone. The domed shapes, the laminated internal structure, the relationship to the ancient seafloor preserved around them, all of it is visible to anyone willing to make the journey. You can look at the earliest record of complex biological activity with your own eyes and understand directly and without intermediary that life was here, doing something organized and persistent 3.5 billion years ago. Similar experiences are possible in different geological contexts, in the Superior Craton of northern Ontario, in the Barberton Greenstone Belt of South Africa, and in the Isua Greenstone Belt of southwestern Greenland, though the Greenland rocks at 3.7 to 3.8 billion years, are so ancient and so metamorphically altered that the evidence they carry is interpretable only through sophisticated isotopic analysis rather than direct visual inspection. In all of these places, the Precambrian is not a reconstruction or an imagination, but a physical surface that predates the visible world. The Proterozoic, the eon spanning 2.5 billion to 540 million years ago, is the era during which the most important biological transitions of the Precambrian occurred. And yet, it is the least studied and least appreciated of the Precambrian eons. The Archaean gets attention because it contains the oldest evidence for life. The Ediacaran gets attention because it contains the first visible complex organisms. The vast middle, 1.9 billion years of Proterozoic time, is the geological era most often summarized as relatively uneventful. This is a significant mischaracterization. The Proterozoic contains the Great Oxidation event, which transformed the atmosphere from reducing to oxidizing, and is arguably the single most consequential abiotic transformation in Earth's history after the formation of the planet itself. It contains the origin of the eukaryotic cell, which is the prerequisite for all visible complex life. It contains the assembly of the boring billion, the period of apparent stasis that was actually the time during which the genetic complexity required for the Cambrian explosion was being built. And it ends with the Snowball Earth events and the Ediacaran biological revolution that set the stage for the Cambrian. The Proterozoic ocean itself underwent a specific and important transformation that is worth examining in detail. The early Proterozoic ocean, immediately following the Great Oxidation event, was what geologists call a stratified ocean. Different chemical conditions prevailing at different depths. The surface layer was relatively well oxygenated because it was in contact with the now oxygen-containing atmosphere. But the deep ocean remained anoxic and sulfidic, rich in hydrogen sulfide produced by sulfate-reducing bacteria, for hundreds of millions of years after the atmosphere began to oxygenate. This stratified condition, sometimes called the Canfield Ocean after the geochemist Donald Canfield, who described it in detail, had important biological consequences. Many essential trace metals, molybdenum, zinc, and others, are insoluble under the sulfidic conditions of the deep Proterozoic ocean, meaning they were largely unavailable to biology during this period. This trace metal scarcity may have been a significant constraint on the evolution of biological complexity during the boring billion, a genuine chemical bottleneck that limited the metabolic possibilities available to early eukaryotes. The progressive oxygenation of the deep ocean during the late Proterozoic, particularly in the aftermath of the Sturtian and Marinoan snowball glaciations, released these trace metals from the constraints imposed by sulfidic chemistry, making them available for incorporation into biological molecules. Molybdenum, in particular, is an essential component of the enzyme nitrogenase, which fixes atmospheric nitrogen into the biologically available forms that all complex life requires. The increased availability of molybdenum in the post-snowball ocean may have dramatically enhanced biological nitrogen fixation, providing the nutrient input needed to fuel the biological productivity of the Ediacaran and Cambrian oceans. The Snowball Earth events, devastating as they were, may have been necessary to break the trace metal limitation that was constraining biological evolution during the boring billion. The Ediacaran organisms themselves deserve one more pass of attention, because recent research has revised our understanding of them in ways that are not yet widely appreciated outside specialist literature. For many years, the dominant hypothesis was that the Ediacaran fauna represented a failed experiment, a group of organisms that left no descendants and whose relationship to modern life was essentially nil. This view was encapsulated in the kingdom Vendobionta concept, proposed by Adolf Seilacher, who argued that the Ediacaran organisms were so fundamentally different from any modern animal group that they deserved their own kingdom of life, separate from plants, animals, fungi, and any other recognized group. More recent analysis, particularly the lipid biomarker work on Dickinsonia and the careful morphological analysis of other Ediacaran taxa, has nuanced this picture. Dickinsonia appears to have been a true animal, not a failed experiment, but an early animal with a genuinely unusual body plan. Some other Ediacaran taxa appear to be related to modern animal phyla, though the precise relationships are disputed. The rangeomorphs, the fractal branching organisms to which Charnia belongs, may be related to early animals in the stem group of a major phylum. Some researchers have identified putative Ediacaran ancestors of various bilaterian groups, the animals with bilateral symmetry, which include most modern animals. What this means is that the Ediacaran fauna was not a uniform failed experiment, but a diverse community that included organisms from multiple evolutionary positions, some that represent genuinely abandoned body plans, some that are early members of lineages that would persist into and beyond the Cambrian, and some whose evolutionary position remains genuinely uncertain. The Ediacaran-Cambrian transition was not the replacement of one community by an entirely different community that appeared from nowhere. It was the selective survival of some Ediacaran lineages and the extinction of others, overlaid by the rapid diversification of lineages that had been present but cryptic during the Ediacaran. The question of why some Ediacaran body plans survived and others did not is one of the most interesting in Precambrian paleontology. The general answer involves his ecological role. Organisms whose ecological strategies were compatible with the new world of predation and bioturbation had better prospects than those whose strategies depended on the quiet, microbial mat-dominated seafloor of the Ediacaran. Organisms that had developed mineralized hard parts for whatever original function, whether defensive or structural, found those hard parts suddenly valuable as armor against the new predators. Organisms with directed motility, capable of fleeing or pursuing, had advantages in a world where movement was suddenly a matter of survival rather than merely a matter of nutrition. The molecular toolkit perspective adds another layer. The animals that survived the Ediacaran-Cambrian transition and radiated into the modern phyla were, in a genomic sense, the most evolutionarily versatile, the ones whose developmental regulatory networks were most capable of being quickly modified by selection to produce new body plans in response to new ecological pressures. The Cambrian explosion in this view, was not the generation of body plan diversity from nothing but the rapid phenotypic expression of genetic diversity that had been accumulating throughout the Ediacaran and earlier Proterozoic. The trigger was ecological: predation, bioturbation, changing ocean chemistry, but the raw material was genetic, assembled over hundreds of millions of years of Precambrian molecular evolution. We are, in the most literal biological sense, the outcome of this process. The human body is built from eukaryotic cells, each containing mitochondria that are the domesticated descendants of Proterozoic bacteria. Our developmental biology is controlled by the same toolkit genes: the Hox genes, the Pax genes, the Sox genes, that were assembled during the Precambrian and first deployed in the Ediacaran or earliest Cambrian ancestors of our lineage. The oxygen we breathe was produced by Archaean and Proterozoic cyanobacteria and is maintained at its current atmospheric level by the photosynthesis of their descendants in modern oceans and on modern land surfaces. The nitrogen in our proteins was fixed from the atmosphere by bacteria whose nitrogenase enzyme contains molybdenum that became available only after the oxygenation of the Proterozoic deep ocean. We are not merely the product of the Precambrian in the abstract sense that all things have causes reaching into the past. We are the product of specific Precambrian events: the Great Oxidation event, the evolution of the eukaryotic cell, the snowball glaciations, the Ediacaran biological revolution, each of which was a necessary precondition for what came next, in a sequence that runs from the earliest chemistry of life to the present moment without interruption. The 88% of life's history that the standard evolutionary narrative tends to gloss over is the part that built everything. The 12%, the Cambrian to the present, the visible world of animals and plants and complex ecosystems, is the part that the Precambrian made possible. The story of life is not the story of progress from the Precambrian to the present. It is the story of the Precambrian, full stop, with a brief coda of 540 million years, during which the products of 4 billion years of preparation finally became large enough to be visible. Go outside. Look at a tree. The tree is a eukaryote, built from cells that contain chloroplasts that are descended from cyanobacteria. The oxygen the tree is releasing into the air is produced by the molecular descendants of the great oxidation events cyanobacteria. The nitrogen the tree incorporated into its proteins was fixed by bacteria performing a biochemical process invented in the Archaean. The soil the tree is rooted in was built partly by microbial communities performing metabolisms that have been operating since the Proterozoic. Now look at yourself. You breathe oxygen produced by organisms whose lineage dates to the Archaean. Your cells are powered by mitochondria that are domesticated Proterozoic bacteria. Your developmental biology was assembled during the Precambrian from genetic components that first appeared in organisms we have not yet found in the fossil record. Your immune system, your nervous system, your reproductive system, all of these are elaborations of cellular and molecular machinery whose fundamental components were invented during those invisible billions of years when the world looked like nothing you would recognize. The Precambrian is not the past. It is the foundation on which everything present is built, and it is still operating. In the microbial world that surrounds and pervades all complex life, in the atmospheric chemistry that sustains the ozone layer, in the nutrient cycles that make complex ecosystems possible. The invisible world that preceded the visible one did not go away when the Cambrian began. It continued. It continues now. Every breath you take is a transaction with the Precambrian. The history of life on Earth is not a 12% story. It is a 100% story, and the first 88% is the part that matters most. Alien chemistry, invisible organisms, orange skies, iron-green oceans, a planet frozen solid and thawed catastrophically twice. The first bodies with symmetries that have never been repeated. The first true animals, without mouths or guts, absorbing their food from an ocean that no longer exists. And underneath all of it, still running, still cycling the elements that make complex life possible. The microbial world of the Precambrian, doing what it has always done on the planet it built. That is the real story of life on Earth. It just requires looking past the visible.